when they come out of the jars of water. And so what we're going to be doing is using a couple of different chemical solutions to try to kind of protect and kind of bulk out those cell walls, kind of support them, so that when we take the water away, the cell walls don't just kind of like collapse down in on themselves and give us this cracking and discoloration on the scales. So some of the chemicals that we use to do that are, this is called PEG, polyethylene glycol. And this is actually the active ingredient in antifreeze. And PEG itself comes in a number of different molecular weights. And what that means is it's the molecule itself can chain. So it kind of chains and you get multiple of them. So the ingredient in antifreeze is called PEG1. There's only one molecule, or kind of one molecule chained to itself. As you get into higher numbers, 400, 1500, 3350, you get more of those chained. And so as it gets higher in there, so PEG1 is pretty toxic. You shouldn't drink it. It's why antifreeze is always kind of, there's big labels on it saying, you know, don't let anybody get at this, and because it's really toxic. But by the time you get up to PEG 3350, there are so many of those molecules chained together that it's not going to hurt anybody. And so therefore you can actually have PEG 3350 is frequently used as a laxative to kind of, you know, help people move things through their intestines. And so it's kind of this really useful molecule. And because it comes in all these different molecular sizes, you can use it for different things, and it kind of will get into the cells better at certain weights. So this is PEG 1500. So that's 1,500 of those molecules chained together. And you can see it's kind of a nice, it comes out of the container in these kind of big waxy crystals, which we then have to dissolve into water, and then we can put things into the water and the PEG solution. This is PEG 3350. That's that really kind of higher molecular weight, and this is actually come as a really fine powder here. It can also come as those kind of bigger waxy crystals. This is just those waxy crystals really ground down. So what we do when we have the cones is we make up our solutions with the PEG. Sometimes we use chitosan, which is ground up shellfish shell. Sometimes we use sugar as well, just sucrose table sugar. And so what we do is we make it up into a solution, and then we put, you know, we have these containers which have these individual wells in them. And so we can put a cone with its clear label right on the container, and we can put different solutions. So, you know, we might have like PEG 1500 in this one, PEG 3350 in this one, chitosan in this one. And that really allows us to try out several different types of chemical. And what we're really looking for is we're trying to determine what's going to be the best chemical. You know, do we want to use that ground shellfish shell? Is that going to give us a better result? Does the PEG give us a better result? But if it does, which weight of it? You know, is it better to use a smaller molecule or a bigger molecule? So this is kind of what we've been doing for the last about year and a half, is we've had our cones in solution. We have then taken them out of that solution. So basically drained the solution out of the containers. And then we have these, you know, we have them still in their containers, still really labeled, because we have a whole bunch of weight and length and all sorts of other data. So we need to keep these actually as each individual named cone. That's really important for us. And so now what we've got them currently is, you know, we've got them out of solution. They're in their individually named containers. And what we're doing is we've got them in a freezer upstairs. Now, normally when we do this kind of technique in conservation, you would take it, if you've got a freeze dryer, you would take the item after it's come out of solution and you would put it into a freeze dryer. And that would kind of quickly take all the water out, and then you could just kind of have the object. Unfortunately, we don't have a freeze dryer, so we've kind of got them in one of our freezers upstairs. And if you think of like, if you have a piece of meat in the freezer for a few years and you take it out, it's got that kind of freezer burn. It's basically been freeze dried. And so that's just kind of over time, the moisture has come out of the cone, or sorry, not out of the cone, out of the piece of meat, and it's dried out, and you've now basically got freeze dried meat. And so that's kind of what we're doing at the moment in the freezer upstairs, is we've got some of our cones upstairs drying, and it takes a long time. Again, if it's a piece of meat you've had in the freezer for several years, our cones have been in there for about 1.5 years and they're still drying. So it's a very slow process, but I do have some of the cones that we did and dried a different way. These got dried just in what's called a humidification chamber, where we basically have a box that we can put extra humidity into and slowly dry things down. So I have a couple of cones here that we have dried already, and so we can take a quick look at them. So this is a half cone that we had, and this was done in chitosan, so that's that shellfish solution. And you can see it actually looks pretty good. Like this is really identifiable. You can look at that and you can immediately say, okay, that's a conifer cone. Awesome. It's not so great. We've got some of these cracks here. We've got a little bit of, this is called sloughing. And this is just basically the kind of top layer of cells have separated from the rest of the cone and are kind of lifting away. That's not really something we want. But overall, that's looking pretty good, actually. And the cones, the scales have kind of opened back up. So that's really identifiable as a conifer cone. Like you look at that and you're like, yes, that is definitely a conifer cone. So that's actually a really good result. We also have some of our sucrose cones. And so this is, again, just table sugar, just regular table sugar. And we did that because PEG is a petroleum derivative. And a lot of conservators are trying to move away from using PEG to conserve waterlogged material, both because it's it's less sustainable, it's less environmentally friendly, but it can also react with things. So for example, PEG can react with iron and create sulfuric acid. So that can be dangerous for people working in museums with those objects. So conservation in general is kind of looking for some different chemical solutions that we can use, more natural solutions to preserve things. So we tried sugar, because that's one of the ones that some other conservators have used. And you can see this one's actually looking pretty good. The colors may be a little different. We do have a few cracks, and we have a little bit of sloughing, but in general, it looks pretty good. The one problem we found when we used the sucrose was that some of our cones crystallized. And so you can see here these white patches here on the cone. That's actually crystallized sugar. I'll see if I can get it close enough for you to see. So you can see this is actually sugar has crystallized on the outside of the cone, and that is 100% not something we want in our collection space. Sugar will attract pests, will attract bugs that will then eat the collections. So we really, really don't want sugar crystallized on the outside of the cones. Also, you can see it's a lot harder to tell what's going on with this cone. You can't really see cracks. You can't see whether there's sloughing. So that's not really a good, that's not a good response. That's not what we want to see. So we've decided we're not going to use sugar. That's a definite no for preserving the actual Snowmass cones. Sugar is out. We're still kind of running some experiments with the chitosan, which is that shellfish shell, and the PEG, the polyethylene glycol. We're still waiting to see for our cones to dry out and see kind of how we feel about those. So that's kind of where we are at the moment with our experiments. I'll probably do another presentation here in the future on kind of what we're feeling, how we're doing, how the project is going. And so that's it for the moment, if anybody has any questions.
Yes, welcome everybody. If you're just joining us to Science Division Live, we're talking about the preservation of conifer cones that we found in a Snowmass dig site right here in Colorado several years ago, alongside other prehistoric creatures like mastodons, and I think even like salamander bones and things were found.
Oh yeah, it's all sorts.
Yeah, and so Kathryn was talking to us a little bit about how we take these very wet and waterlogged specimens and then conserve them in our collections for study. If you have questions, feel free to put them into the comment section. We'll try to answer as many of those questions as we can. I have a quick question for you, Kathryn.
Yes.
what is the risk of allowing these specimens to dry out too quickly? I mean, presumably it should be easy to dry something. Why is it such a long process to dry them out? And what happens if they dry out too fast?
If they dry out too fast, we get all that cracking and the splitting and the kind of peeling away of the top layer of cells. And so we found with the cones, but also in general with like pieces of wood. So conservation has been preserving waterlogged wood for like a good 50, 75, nearly 100 years now. And some of the very earliest pieces that came out of like excavations in Europe, they'd find ships either buried on land, but in kind of like waterlogged, like boggy areas, or it's warships that sunk during the age of sail. The warships would go out, they'd sink, and then they were found in the 1950s and brought up from the bottom of the sea and trying to preserve them. And this is a thing that we found on sites where material has been waterlogged. If you bring it up and you just like, for example, you have a beautiful like carved piece of wood. If you bring it out of the water and just kind of like sit it on the side of the trench for a couple hours and let it dry out naturally, it will dry and crack and kind of like twist on itself. And what was like maybe, for example, a beautiful cone will suddenly be completely unidentifiable just because of the way that the cells have kind of collapsed under themselves. Because if you think of cells in a waterlogged piece of like wood or leather or that kind of thing, if you think of like a water balloon that gets overfilled and it's right at the point where it's about to burst, if you take all the water out, it just kind of like collapses down on itself and looks really sad. That's the same sort of thing that's happening there. And you've got the added processes of like bacteria eating at the cell wall to weaken the cell wall. So basically, if you think if you think of a waterlogged cell as being like absolutely full of water, if you take all of that water away, it just kind of like collapses down on itself, and that causes cracking, twisting, fracturing, flaking, all kinds of things, which basically makes the wood completely identifiable. And we're seeing that same thing with the cones. And so that's kind of the real hazard of just kind of like air drying things. If you don't use the chemicals to support it, you don't use the you don't kind of bulk them out, you don't carefully dry them out, you're going to wind up with something that you can't use. It's completely unidentifiable.
Interesting. It's something you never really think of. In the movies, they pull something off the ocean floor and then they stick it in a museum 2 days later. Obviously, that process is a lot more complex.
Yeah.
One of the questions we have from the chat, from the comments is, what can these pine cones teach us? Why go through all this effort to preserve pine cones? And what can they teach us about this era, this time and place?
Okay, yeah, that's a great question. And it's these are actually, the Snowmass site is about 70 to about 130,000 years old. So these are what are called Pleistocene era sites. It's a Pleistocene era site. And the Snowmass site itself is actually the only high altitude site in North America that we have. So it's the only alpine site. So again, Snowmass itself is just up next to up to Aspen. So it's way up there in the mountains. So this is the only high altitude site that we have in North America. So first of all, like, it's a unique site. So these cones are super important. The other thing that makes them important, besides just their age and the fact that it's the only site here in North America, is that these cones are helping us to identify and understand what the environment is like around the site at that time. So it's two different periods, the 70,000 years ago and the 130,000 years ago, that the site was really occupied by plants and animals. And it's slightly different species of trees. And so we know kind of what the environment those species of trees live in today. So we have a really good idea of what the environment was like at the time that these cones were pulled out of the ground. Excuse me. And so this is actually really, really important. Being able to study these cones actually gives us a really good idea what the environment was like at the time, kind of maybe what animals are living there, because we know that certain animals eat these materials or eat things that are found in the same forests as these trees. So it's a good indicator of the environment at the time. It's a good indicator of what's going on with tree species, what's going on with the climate, and kind of what's really happening. It helps us also to, like, if you've just got the animal bones and that's all you're interested in, you actually lose out a lot of information about what's going on around the site at the time. So that's why these are so important. And when they're just in the jars, you can't really look at them. You can't really get a lot from them because, you know, if you just think of something that's held in water, you can't get as much information from it as you can from something that you can hold in your hand, you can investigate, you can put it under a microscope and look at it. So that's kind of really where and why these are so important.
Yeah, you know, I guess it's when we see fossils displayed, they're usually by themselves, right? It's like a dinosaur skeleton or anything else, but fossils really are an example of how nothing in nature exists in a vacuum. All creatures are interacting with each other. They're eating. They're part of an active ecosystem. So I guess studying every part of that ecosystem is important. We also have some questions in the comments about the physical processes of freeze drying. Why is freeze drying such an important step in this?
So freeze drying is, so like I said, you can kind of do the same thing slowly in the freezer. Freeze drying itself is important, because it's faster. So it means that you're not holding, you don't have a lot of material in the lab all the time. The other thing that's important about it is it's a little bit of an easier process than just slowly drying it in the freezer. Because of course, when you're slowly drying something in a freezer temperatures can fluctuate over time. Like for example, we had about a month or two ago, there was a fault in our freezer and it was kind of getting above the temperature it should have been. And so that was we had to move the cones and everything else that was in the freezer and basically get the freezer fixed. So that's one of the problems with just slow drying it is, you can have issues with temperature fluctuation in the freezer. Another reason that it's super helpful is that it's a little bit more of a controlled process. So what happens in a freeze dryer is you put the item in and then it can either be pre-frozen or there are some vacuum freeze dryer. And so it'll get down to a very cold temperature and then it will go under vacuum, which means that it's got a pump, which is basically pulling all of the air and all the moisture out of the chamber. And so as it's under vacuum, it will then heat up, which then causes the ice. And this is basically, it's a chemical feature of water. If you heat it fast enough, it goes from ice. So if you think of water's three states, liquid, ice, and water vapor, when it's in the freeze dryer, as it starts to heat it up, it heats it up fast enough that the ice goes from ice to the water vapor. It doesn't go back through that water phase. It just goes straight from ice to water vapor. And that means that it's easier to pull out of the cones and you don't have the problem of, water expands when it freezes and then it kind of shrinks and then it kind of expands again. And basically you keep it from going through a couple of those phases where it can cause more damage to the object. So freeze drying can actually cause less damage than a slow freezing over time. And it's just, again, it's a more controlled, and you do that through several cycles until you've got all the water out. And it's a more controlled, more precise way of getting water out of objects, which is why it's preferable to slow drying.
Yeah, I didn't think about that sublimating would get rid of some of those problems. I have one final question for you, and that is, what are some of the unique challenges that these Snowmass cones have presented to you that you haven't encountered in other situations?
So the cones are a challenge because so most conservators handle waterlogged wood or other plant material, baskets, leaves, that kind of thing. Cones are not something that a lot of people have tried to conserve. We couldn't find anything in the literature when we started this project. We were kind of looking and saying, okay, well, what have other people done? And basically not a lot of other people have tried it. And cones are difficult because they're wood-like, but they're not wood. So their cellular structure is different. So if you think of a piece of wood, you've got those kind of like nice, long, thin cells, which kind of like form, if you think of like a tree trunk, it's got those long, thin cells, the xylem and the phloem, which are kind of really pulling moisture through by capillary action, all those kinds of things. When you put that into solution, it holds the water, it pulls the solution in the same way. Cones don't have that same sort of structure. They have, let me see if I can find, so they have a cellular structure like this. And so they've got lots of kind of like small sorry, I can't let go of the camera because my stand has suddenly decided it doesn't want to stand up straight. So it's got all these kind of small cells, and it's got some cells that are aligned in those kind of like nice long thin bands, but most of the cells are kind of more a honeycomb pattern, but not as regular. And so you'll get things like you have this nice long, doesn't want to focus on this now, but you'll have this kind of like nice long central channel, and then it kind of goes out into the sides, and then each of those scales has this kind of really all over the place cellular structure, which means that solution doesn't get into those cells as easily. It's not kind of naturally pulled in through capillary action, which makes it really, really difficult to get solution into them. It makes it really difficult to preserve them. So that's kind of one of the big problems that we're facing with this project, is just getting the solution into the cones. Some of the other people we've talked to have had problems where, you know, they're like, I followed a standard conservation treatment. I put it in this solution for the standard amount of time, and I wound up with like a crust of the solution around the cone instead of, you know, anything in the cone. And we've actually seen that with some of our cones as well. Some of our cones formed crusts as well. They're some of the ones up in the freezer. And so, yeah, it's a real problem. And it's just the cellular structure of the cones is making it really, really difficult to actually get the solution into the cones. And so that's kind of where and why we're going through and we're trying so many different solutions, because we knew this was going to be a problem. As soon as we learned that it doesn't have that same kind of cellular structure as wood, we were like, okay, this is going to take a completely different sort of approach to do this.
Wow, you know, things you don't think about, you know, like the challenges because of the cellular structure of a pine cone for its conservation. And I feel like this is, you know, a glimpse into the incredible work that conservators like you do to really enrich and deepen our understanding of the ancient past. So I have to say, you know, thank you so much for sharing what you have been working on. I know you said that there'll be updates in the future, so I'm sure we all would love to hear how this work is going. I want to thank everybody who's joined us, who's tuned in and joined us. I hope you've enjoyed the program as much as I have. Thank you for your wonderful questions and comments, and we hope to see you again soon. Thank you, Kathryn, and goodbye, everybody. Thank you so much.
Thanks. Bye.